So just so you know you're in the right place, this is breakout session 1, genetic diagnosis, why and how. So if that's not what you're looking for, the other breakout sessions are across the hallway. So, all right, so my name is Laura Rufabach. I am the co-president of the Jane Foundation. In the Jane Foundation, we specifically work on LGMD2B, but today we're here to talk about how to get a genetic diagnosis and why it's really important. So to start, I'm just going to give a brief overview of the topic, and then each of our speakers will go into a different aspect of it in a little more detail, and then the goal is at the end we'll have time for question and answer. It is a big room and there's no mics out in the audience, so you'll have to yell your questions, or you can come and bring them up to me and I can help facilitate that. So first I wanted to start with what is a diagnosis, because that can mean a lot of different things. And so one, there's a clinical diagnosis, and that's how most of you would have started, is you go to your physician, your neurologist, and they give you, based on what they see about your muscle weakness, where the pattern is, other laboratory tests, and they determine what they think you might have based on those things, and so that's a clinical diagnosis. Then there is what I'm terming a molecular diagnosis, and this is usually based on things that are not genetic testing. They're based on, like, they've tested your muscle, a biopsy to see whether it looks like it's a muscular dystrophy or something else, or are you missing a certain protein? So that I consider a molecular diagnosis. And then we have the genetic diagnosis, and this is based on identification of disease-causing mutations found in your DNA. And so this really tells you what gene is malfunctioning that's leading to your disease, and then identifies the specific changes that are causing the problem. And that's what we're going to talk about the majority of today. So the question is, well, why do I need a genetic diagnosis? I've been told I have a limb-girdle muscular dystrophy. There are no treatments. Why do I need a specific genetic diagnosis? So the reason is there's a bunch of different reasons. The first is it tells you what the pattern of inheritance, and that means whether it's dominant, meaning you can pass it directly down to your children, or recessive. And so understanding that will help you know what other family members' risks would be. It also can provide you information on how to manage your disease better for yourself. Can answer questions, because the different LGMDs have different risks. Some have cardiac risks, some have respiratory risks, some progress faster, some progress slower. So it can tell you, will my heart be affected? How fast will it progress? What muscles will be affected? And are there things I should be doing or should avoid? And lastly, knowing your subtype also helps you be able to be in clinical trials, because when you have a clinical trial, it's typically on a specific subtype. And so in order to be eligible, the trial is for LGMD2B. You have to know, do I have LGMD2B? And the study coordinator is going to want to know, well, what mutations you have, and to confirm that. So you really need that to be able to participate in those studies. So how do I get a genetic diagnosis? The panelists will go into a lot more detail, but there's a lot of different types of genetic diagnosis, and the tests are not all the same, and what they can identify is not all the same. So you need to kind of understand, just because you say you've had genetic diagnosis, have you had all the types that are needed, or especially if you haven't had an answer, there may be other things that you could try. And so you may need more than one type of analysis in order to get your answer. And I want to stress that even if you have genetic testing, not everyone who gets genetic testing gets a confirmed diagnosis after that. Sometimes it takes more, and one of our panelists will really go into more about that. And as people have talked about, there are a few genetic testing programs that are currently available. And there is some testing that people are collecting samples for here. And I have flyers up at the front, so after the session you can come up and get them. That gives you information about where you could get some testing. So we have 4 great panelists for you today. And they'll be each making a small presentation, and then we'll have the question and answer. We have Rich Dineen, who's a certified genetic counselor from Rush University Medical Center here locally. Then we have Dr. Peter Kang. He's an MD out of the University of Florida, Gainesville. And then we have Dr. Conrad Weihl, who's an MD, PhD, out of Washington University in St. Louis. And then we have Dr. Monkol, who is a PhD out of Yale University. So first up is going to be Dineen. So sorry.
All right, good morning, everybody. Hopefully, is everyone able to hear me okay? All right. So just as I explained, I'm based here in Chicago, and I'm a genetic counselor at a medical center called Rush University Medical Center. And how do I advance this? Right here? Okay. All right. And so first of all, I apologize from some of the other speakers that there may be some overlap on the few things that I'm going to talk about. But, you know, what has really been changing in genetic testing? And I've worked in a neuromuscular clinic maybe about a decade, and a lot has changed from how we used to do testing. I think we've already kind of touched upon some of the importance of genetic testing today, that we think it's really pretty important that people have the testing. But also answer some of the questions, the typical steps to the genetic testing process, because there's a lot of misconceptions. And I think a common theme is, or misconception is, that genetic testing is expensive, and it no longer has to be. The one thing is, I probably should have used the word can. Genetic testing can provide absolute confirmation of a diagnosis. There are, lots of situations where testing results may be less clear. A lot of the different treatments today are definitely being designed either with a specific gene in mind or based on a specific genetic change within a gene. But also stressing that positive results are often necessary to participate in a disease registry or to qualify for a clinical trial. And so this slide is really meant for those of you in the audience, or a lot of you actually interact with people worldwide who may have a diagnosis of limb-girdle muscular dystrophy, who simply have a clinical diagnosis, who may have pursued genetic testing many years ago. I think we've already stressed that in the past we used to have to test a single gene at a time. It was cumbersome and it was expensive. But that genetic testing panels where we test groups of genes together is where we start today. Some people have concerns, well, I need to go through another muscle biopsy, or, how do they actually collect a specimen? The thing to also sell is most laboratories today can do genetic testing off of a saliva sample or a mouth swab sample, so usually not even a blood draw is required. And the cost has drastically changed through genetic technology. In this slide, I'm just highlighting, there's no need to really remember the names, but just some of the differences in techniques over time that have helped to reduce the cost and increase the ability to actually find an answer. So in the past, we were using a testing called Sanger sequencing, which was more cumbersome, more costly. And then several years ago, the development of something called next generation sequencing, which was really that technology that allowed us to test for multiple genes at a single time and help drive down the cost. But it's also important, not everybody with their past genetic testing, a lot of times we'll talk about sequencing, or basically the spell check method to look at a gene. But that method doesn't really look for large missing pieces or extra pieces of genetic instructions within a gene, and that requires a separate kind of test. And so that's a test that would maybe look at what we call copy number variants. There's also other methods that are being developed today, such as RNA sequencing, for those individuals who may have had testing and maybe didn't receive a complete answer. Just in terms of the steps of the process, that the neurologist typically would pick the category of testing that needs to be performed. I also want to stress the sample can be collected right at clinic, or increasingly so if there needs to be some sort of investigation in regard to costs, a sample can be collected at a later time. Many laboratories today have ways to look into healthcare coverage to see if the testing would be covered or not, what the out-of-pocket expense might be to a family. And then I think it's also been stressed, some of the laboratories out there have had fairly low cash options around the $250 mark. And that results typically take several weeks to come back. We're not actually going to be talking about test results. There's a separate breakout session that I just sort of wanted to promote in the Genetics 101 in breakout session number 3. I am a big believer, though, in people having an individual of their genetic test results. Maybe a show of hands, how many people actually in their possession have a copy of their test results? So not as many as I'd like to see. So in those things that you want to know is, are my results indeed positive? And what types of genetic changes might I actually have in my genes, and what do those tell me? Some questions to maybe ask your providers that work with you is, from your testing report, how long ago was my testing performed? What was the specific laboratory that did my testing? Was it a research laboratory? Might it need to be repeated somehow? How many genes were actually contained in the actual panel, if I did have a panel, that were analyzed by this testing? What laboratory method was actually used to look at the genetic changes? And did my testing provide an answer, or might I be somebody who could benefit from some more current testing? Sharing results with other family members is important, not only for family members who may have similar symptoms so that they don't undergo the same diagnostic odyssey, but also for things like carrier testing for other family members, things of that nature. But in a practical sense, doing targeted testing is also a lot less expensive than sometimes doing a broader type of genetic test. And a common question families in clinic will ask is, how is genetic testing covered? Again, many laboratories today are able to do a benefits investigation to try to determine what the out-of-pocket expense may be. And that decisions regarding genetic testing can also be made outside of clinic if someone's feeling, do I need to make this decision right now? There's ways to sometimes make that decision after clinic. But I also want to stress there's also a number of no-cost options to the LGMD community. And I'm going to list 2 here, one being the Lantern Project, which is a project that's sponsored through Sanofi Genzyme, and tests for approximately 100 genes in testing that's performed through a commercial laboratory called PerkinElmer Genomics. And then another commercial laboratory called Invitae just launched a testing program called Detect Muscular Dystrophy, and also has testing panels that include genes for LGMD. And I think that is it for my piece, and I'll turn it over to the next speaker. Thank you.
So I'll kind of pick up on the theme and keep going with that and touch upon research a little bit also. So these are my disclosures and relationships. So as a lot of you know, there are a lot of genes for limb-girdle muscular dystrophy, 36 plus and counting. And some of you may have also heard that the classification system is changing. So a lot of you who have genetic diagnoses know of yourselves as having type 2B or 2C, etc. What happened was, you may have heard, we ran out of letters. We got to 2Z, and so a group got together in 2017 or 18 and proposed calling the dominant ones D and then followed by a number, and then R for the recessive followed by a number, and that's being adopted as we speak and is going to become more widespread. So I think most of us are familiar with both types of systems, so you don't need to worry about it, but that's what's happening in the background. And most limb-girdle mutations to date are small nucleotide variants or other small changes, and thus they should be picked up on various sequencing platforms, as Richard mentioned. So Richard mentioned some of this already. I'll go through it quickly. Why do you want to get genetic testing? You want to put a name to the disease and have some certainty, and that's something that's a really powerful bit of knowledge for you to have. It helps you join the community in a little bit more of a structured way. And as you know, there are organizations that are dedicated to specific subtypes of limb-girdle. The different subtypes are associated with different complications in clinical courses, so that's one way it could benefit you to know your diagnosis. And then eligibility for clinical trials and novel treatments. There are new treatments coming out for various diseases now that are gene-specific and sometimes even mutation-specific. And then for genetic counseling and family planning purposes, as Richard mentioned. So I think one thing that I'd like to help sort out is the difference between clinical testing and research testing. And Richard talked a bit about the clinical genetic testing. And so back years ago, we used to do what's now called Sanger sequencing a lot, where you go through one gene at a time, or if you combine those into a panel, it was really expensive. And it's been really nice the last decade or so to have next generation sequencing available. And on a clinical basis, the panels that you hear about that are targeted towards limb-girdle, for example, they're usually based on what's called targeted sequence capture. They only sequence those particular genes. And then there's exome sequencing, where you sequence every gene that's known, and there are pluses and minuses of both types. CLIA certified means that there's an agency that certifies genetic testing laboratories and makes sure that they meet certain quality standards, their accuracy, et cetera. So that's a little bit of a safety net for patients who get clinical testing. And then usually as part of this, you get results within a certain time frame unless there's an unexpected technical issue. And then you get an official report. And as Richard said, I think it's very important if you do have a genetic diagnosis, you should ask for a copy of your report. Now, research studies, in the clinic, I order clinical genetic testing. On the research side in my laboratory, we also try to dig a little bit deeper for especially those patients who don't have diagnoses. So that we do use next generation sequencing, but we focus more on exome and genomes. And then we primarily use Sanger sequencing just to confirm mutations. We're not CLIA certified because we're a research lab. There's no time frame. And we're not allowed to report results directly to families, but there's an indirect way for anybody who's interested and wants to sign up for our study, which is in the Atlanta room. We'll explain details, but there is an indirect way for you to get a report through your physician, but it takes us a couple of steps. The problem that we've come up against, and I think some people in the audience may have come across this personally, is that clinical genetic testing can't diagnose everybody. And there is a recent paper published where they looked at thousands of patients that had these panels done, and the diagnosis rate was 27%. Now those thousands of patients may not have all completely fit the limb-girdle diagnosis well. So I think it may be even higher than 27%, but it's somewhere in that ballpark, somewhere between 27 and 50 is my guess. And then in our research lab, when we take those patients who don't have a diagnosis and we look at those, we've been able to diagnose between 20 and 50% depending on the cohort. And then what we'd like to do in the corner is one of my former postdocs, Hema Kumar, who helped a lot with these projects. So when you go beyond standard exomes, you can look at reanalyzing some of this data using advanced bioinformatics techniques and then novel sequencing technologies. So some of the novel approaches we try to use, and this is Christine Bruhls, who's in the audience right now, and she's helping us out, is we can look at transcriptome sequencing. That was something that was published by a collaborative lab by Daniel MacArthur's group. And then Christine developed this, this neat pipeline for looking for cryptic splice sites. And then we also want to try to look at structural variants, which are the larger mutations using various techniques. So take-home points, genetic knowledge has really exploded in recent decades. Many genes have been described, and a lot of patients now have genetic diagnoses, which is really wonderful in terms of getting to know your disease. Obviously, we'd like to see more treatments and hope that that will come. The thing that we want to keep in mind is that many patients do remain undiagnosed, and a negative result on your genetic testing doesn't exclude the diagnosis, so that's only part of the puzzle. And for those who do participate in research, it does help us a lot, and we're very grateful. You've got this flyer in your packets, but if anyone is interested in our study, we're based in the Atlanta room. We're going to be open today from 12:15 to 3 o'clock. And we should be able to stay a little longer in case there are more people interested. And then tomorrow we've got different hours open that are in your packets. The only thing that's a little bit of a limitation, several of us will be heading for the airport at right at 2 o'clock tomorrow. But other than that, we're flexible, and the other sessions we can stay a little bit over if we need to. So feel free to approach us anytime, me or any of my colleagues. And thank you very much.
Great. Thanks to the previous two speakers. So I'm going to kind of talk about a workflow about how we think about genetic testing. I'm a clinician, and I think we need to start thinking about how basic scientists or lab scientists, geneticists, and clinicians, and how we need to work together in order to make a genetic diagnosis. And so I just turn that off. I think I do that. So I want to talk about genetic testing just very briefly, about some of the barriers to genetic testing: payers, community knowledge, and the ultimate one that I think is most important is test interpretation. We can do a genetic test, but if we can't interpret it properly, if we don't understand how to interpret it, then we really have not done genetic testing. We haven't done any service to anybody. The evidence-based guidelines actually in 2014 were actually, this is the most recent for clinicians, so for neuromuscular physicians, actually was level B and level C evidence. It was actually level C evidence to do exactly what Dr. Kang just talked about. Level C evidence means that it's just expert opinion. There's no evidence to say that it actually changes people's lives or anything. Level A, of course, is the best. And so in 2015 to 2018, the Muscular Dystrophy Association as well as the Jane Foundation actually were able to actually perform free genetic testing and remove that one barrier, which was who's going to pay for it. So they removed that barrier. They sequenced 35 LGMD genes in about 3,000 patients, and in aggregate with the Jane Foundation around 6,000 patients. And so this is a representation here of the genetic kind of architecture of what they found in patients with limb-girdle muscular dystrophy. And this is great. This shows that 20% of patients with a genetically defined limb-girdle have, for example, LGMD2A. However, what this doesn't show is this next slide here, that was only 25% of the patients. So of the patients that actually got genetic testing, that doesn't mean they don't have limb-girdle muscular dystrophy. They have the phenotype of limb-girdle muscular dystrophy. It means that they weren't able to be solved by a genetic diagnosis. So almost 77% of patients who've had genetic testing for limb-girdle muscular dystrophy are walking around without a definitive diagnosis. And that places patients in a real diagnostic limbo of how do we solve that. So I'm going to talk about the 50% that have an unresolved variant of unknown significance, and I think Monkol will probably talk more about negative gene testing patients. So the way that we do genetic testing is that we sequence every single gene in the genome. So we sequence every single exon. That's one way to do it. And so if we sequence that, as we filter down, and so I don't have a something to point, but as we filter down and we get to that 3,000, everybody has a variant of unknown significance. And what does that mean? That means that if I get sequenced, I have 3,000 variants of unknown significance, 3,000 mutations in genes that a genetic test company would probably say is a variant of unknown significance. So we all have these, not patients just with limb-girdle muscular dystrophy, but everybody has variants of unknown significance. And so we don't all get genetically sequenced, so we don't know that, but we all have these variants. And so what I wanted to do is kind of show, go through a bunch of genetic test reports. So you can see this is an example of a test report that we get, where we get, if you look on the very right side of the screen, you can see, so the left side has the gene, the right side has the classification by the genetic test company, likely pathogenic, uncertain significance, uncertain significance, uncertain significance. This is what your physician is getting. And you think you're confused about what your diagnosis is? I'm confused about what the diagnosis is. So this is very challenging to understand. Here's another example of a patient. One of them is pathogenic, uncertain significance, uncertain significance. These are huge challenges for clinicians. And what's happening is that patients are getting genetic testing done first. So they're going to their doctor, they're saying I have weakness, they're going to a community neurologist, and they're getting genetic testing first. And a community neurologist can't understand and shouldn't understand. I can't even understand sometimes what these are telling me. Again, on the right side, uncertain significance, uncertain significance, uncertain significance. Again, here's one that's pathogenic, but I can't find two. So for this gene, I need actually two mutations. So my point here is not to obsess about these, but just a point that you're not alone if you have genetic testing and it says uncertain significance. So I wanted to give this one as an example of how we think we can throw things over the finish line and actually make that diagnosis. So this is a patient here with, in the top, has two mutations in a gene called calpain-3. Calpain-3 causes limb-girdle 2A. You have to have two mutations. You have to have gotten one mutation from your mom, one mutation from your dad. And in this patient, he's got one mutation that says it's pathogenic, and the other says a variant of unknown significance. So as a clinician, I need to know, does this patient have limb-girdle muscular dystrophy? More importantly, is this a patient that I'd be able to enroll in a clinical trial? And even more importantly, is this a patient that I could give a disease-modifying therapy to because I have made and established a diagnosis of limb-girdle 2A? This patient is in diagnostic limbo right now, and we think that we can throw them over the line. So I'm going to give you his scenario here. He's a 35-year-old man with limb-girdle muscular dystrophy of unknown cause, and his panel shows exactly what you see here. Shows a variant in dysferlin, which causes limb-girdle 2A, and shows two variants in calpain-3, one of which is reported as pathogenic and likely is pathogenic. And so what we then do is we do an algorithm that we think we should start going through, which really needs to be done by the clinician. You can't ask the genetic test company to do this algorithm because it needs information like the patient history. What was his age of onset? What were his first symptoms? Does he have cardiac problems? Are there skin changes? Things that can correlate with other types of limb-girdle muscular dystrophy. His history of needing a cane or a walker in the family. So is there a family history? Maybe not a family history of limb-girdle muscular dystrophy, but a family history of needing a cane or needing a walker. Are there cousins with weakness? That can be challenging. Not everyone knows their family history, and so these are challenging things. A patient physical exam, a really good exam that characterizes, do they have proximal, distal, symmetric weakness, the types of weakness that we think about as clinicians to try to integrate that genetic test information. So in this guy, his weakness was at 25, he had difficulty climbing steps. No family history of weakness. He's got proximal weakness. He has something called scapular winging. So he's got weakness in his shoulder girdle, and he's got something called scapular winging. That's a distinctive feature that can be seen in some muscular dystrophies. It's not diagnostic, but it's a helpful feature. The next issue for variant resolution is to integrate then laboratory data. What's their muscle enzyme? So their CPK. Have they had an EMG done, an electrocardiogram to check their heart? Have they had an MRI done of their muscles? And most importantly, have they had a muscle biopsy? And this is important. So you may get genetic testing, and you may still require a muscle biopsy. And I think that's important to know. You may get genetic testing done, and a muscle biopsy will still need to be performed. And so in this patient, his CK was modestly elevated, 2,000. He had an EMG that showed that he had myopathic changes, that there wasn't evidence that this was due to a neuropathy, helpful. His MRI looked like this, where he had fatty replacement in his posterior compartment of his calves, and he had a muscle biopsy, which showed that he truly had a myopathy. And then you want to do bioinformatics, so database searches, look for functional predictions. Some of this is done by genetic testing companies, but some of it's not always done. And so in this case, this calpain-3 variant has never been reported. It's actually in a splice site area, so it potentially could be pathogenic. And this dysferlin one is rare. It's been reported in the population, but it still has not been unreported. And so those things are helpful. And then finally, we need to integrate other genetic and functional data. So has this patient had an expanded pedigree? So we're actually able to contact this patient's family and see if his parents are alive, see if his siblings are alive, so we can understand maybe those mutations actually weren't inherited on two chromosomes, but are on the same chromosome. And do other tests as well, particularly looking at their muscle biopsy. And so this is an example where we were able to look at his family. These plus minuses show that it likely was inherited at least on two different chromosomes. We were able to look at dysferlin and rule out that it likely wasn't due to loss of dysferlin, and we're actually able to show through another test that he actually had an absence of a protein called calpain-3. And so we were able to diagnose this patient with limb-girdle 2A. And so while that's helpful for the patient and tremendously important for the patient, how can we then go even do better? How can we then deposit this into databases so that people know? And so there's a database called ClinVar, which can help you identify those things. And we're working with a group called ClinGen to kind of develop this kind of engine that can help deposit these things in. So we have sharing of data, and we know that the data has gone through that rigor that I just described. The reason that we should do variant resolution is genetic test results are complicated. Variant resolution doesn't just rule in a variant. It doesn't just tell you have a diagnosis. It now tells you that dysferlin variant likely isn't pathogenic. So it rules out variants. And I showed you all those variants of unknown significance. Potentially we can rule those out. It requires an integrated process of history, exam, family history, testing, biopsy, and other genetic data. And so we heard about GRASP LGMD earlier today, Nick Johnson. This is part of what we're doing as part of the pathway for what we call variant resolution and working with ClinGen. And here's my group of people that I work with who help with it, but I'm excited to answer questions a little later. Can I help you? You got it?
So I'm the last speaker on this session. Can everyone hear me? I don't know if this mic's on quite loud. Otherwise, I'll just talk very, very loud if you can't hear me. So yeah, thank you for the organisers for giving me the opportunity to talk. And since I'm the last speaker, I've got to mix it up a little bit. So I'm going to give you one exciting why and two very brief hows. And the exciting why, the why is very important because I've been told some of you patients don't actually turn up and actually give your DNA. So I'm really going to have to sell on the why. Why actually do this? And so I'm going to use my story. In 2011, I got a genetic diagnosis. So Laura talked about the difference between a clinical diagnosis and genetic diagnosis. So although I had a clinical diagnosis in my early 20s, it took a long time to get that genetic diagnosis. And that was because, as our first speaker was saying, they were using older technology where they were sequencing one gene at a time. And so why my gene was sequenced quite late in that one gene at a time sort of like process is because it was actually really rare in Caucasians, but you can probably make that conclusion I'm not actually Caucasian. So it's very important, I know there are international guests here also, it's very important to know that although some of these genes may be more prevalent in particular ethnicities such as Caucasian, it may not be the same for people in South Asia, East Asia, and things like that. And it's just for the clinicians to know that in the audience. So this is how a genetic report looks like, as a lot of you know in the audience how it actually looks like. And and this is zoom in on the details of the exact mutation. So now I'm going to touch upon the why. Why get a genetic diagnosis? And one of the big reasons why is so you can actually move towards therapy, because the first thing a patient says soon as after they see a genetic report is what now? Would be the question, rather than wow, this is all I wanted. It's the what now? What do we do now? So I'm going to now talk about the translation, the why. Why should you be so excited right now? So when I arrived when I came from Australia to arrive in the United States, there wasn't a lot of technologies around to actually do something about a genetic diagnosis. And it's only been the last 2 to 3 years that gene editing technologies such as CRISPR have come online. So this allows us to go to the human genome with great precision and actually change it and really realize personalized medicine. Based on your genetic mutation in your genetic report, we can actually do something about it. And this is me giving a skin biopsy in 2016. So skin biopsy is the smallest thing. So this was at a University of Massachusetts medical school. So this is me giving a skin biopsy, and then after a few days later, this is the skin cells growing from my skin biopsies, and this is a conversion to muscle cells. So the green represents a protein that's specific to muscle that lights up, and the blue represents the nuclei. And so the elongated shape is how typical muscle cells look like on a dish. So that's giving the skin biopsy. And by giving cells from yourself, this is where researchers can actually try some of these gene editing techniques. And so in 2019, 1 week before my 40th birthday present, I received the best birthday present ever. So So my collaborators who took my cells back in 2016 were able to correct the mutation in my gene, TCAP, that was actually causing my disease. So I can't point to it, but it was an 8 base pair duplication. They designed a technique to go directly to that and actually correct and collapse the 8 base pair duplication, so so my cells could actually produce the muscle protein that it couldn't produce. So this was at 80% accuracy. So they published it in the most, Nature is the most prestigious journal you can actually publish this result. So this shows from genetic diagnosis what can actually happen with the technologies that have only have come online in the last 2 to 3 years. So, and this is very personalized. This technology that they actually had to develop was actually very specific to this patient mutation. So although I had a very rare form of limb-girdle muscular dystrophy, there are research processes out there that can actually look into actually correcting that. And so just to remind everyone, for those who have a genetic diagnosis and those that do not, it always starts with a genetic diagnosis. So the catch is, to precisely edit your mutation, we actually have to know what the mutation is that it's actually causing your disease. So I'm going to touch very briefly on 2 of the hows. So we have this great model here who's actually featured at the back. He may be taking autographs at lunchtime for one of our projects, the Rare Genomes Project, that is also in the handout. The way the Rare Genomes Project is a little bit different to the other projects is this is whole genome sequencing. So this is really for people that have gone through panel sequencing and we haven't found an answer. So it's the hope that for a very small subset of those, whole genome sequencing, so looking at the 99% that I think Dr. Wicklund touched upon, that we can use some of the advances to actually interpret some of those variants in the 99%. And because this is some of the reasons why we don't have a genetic diagnosis for those patients. And another is a research study happening at Yale, so called Seeking the Gene. Again, it's very similar to a lot of the research studies that are happening at the moment. And Dr. Kang had actually talked about the differences between finding a discovery in research setting versus the clinical setting. So this is a research a project that's running out of Yale, and we're interested in recruiting patients also. So it's a great time to be a patient looking for a genetic diagnosis, because there's plenty of research projects out there trying to help you find a genetic diagnosis. Thanks.
All right, so we have about 7 minutes for questions. I have a whole list of questions if you don't, but I will open the floor, but you'll have to talk really loudly so that the panelists can hear your questions. Raise your hand. Yeah.
You guys have mentioned several barriers to genetic diagnosis, one of the chief ones being cost, and it sounds like that's being overcome, so that's great. Another barrier that I know of is discrimination practices in the U.S. I've heard of several folks with different types of muscular dystrophy, including limb-girdle, who don't want to be diagnosed because of, say, life insurance for a spouse type of thing. Can you guys think of any other barriers of why folks are not wanting to come forward to that, to kind of make it more aware in this session for different folks of maybe some of those barriers that you've seen in either in your clinics or in your academic centers, or even interacting with limb-girdle muscular dystrophy patients?
So I mean, I think I can name a couple of barriers. One is knowledge of the patient, to actually know that they can get a genetic diagnosis and actually need a genetic diagnosis. I think that information doesn't always get out. We only see the patients that I can see in clinic. And if there are patients out there that have challenges with access to care, challenges with getting to physicians, that can be quite challenging. And so actually the GRASP LGMD consortium is trying to look at some of this by doing telephone video kind of conferences with patients, but that's one challenge. The other challenge is knowledge of community physicians. So if you think about people's diagnostic journey, it's often, they're told that they're just getting weak. I'm sure many of you know this story. You're told that you're getting weak, it's your back. It's, so you go through, so as far as a barrier, thinking about how can we start pushing patients further, and if they have an elevated CK, for example, that genetic testing may be something that needs to be done sooner. And so I think consensus guidelines that don't say that it's just my opinion that you should get genetic testing, that it's standard of care to get genetic testing, I think is one example I can think of.
Yeah, I guess another barrier that some people encounter is not having a clear knowledge of the benefits. Even we do hope to come up with more treatments, but even when there are not, there's a lot of power to knowing your diagnosis. It has implications for your family. So it is very important, in my opinion, to try to get as much information as possible. So that's one of the things we're trying to promote.
I think family education is another barrier. It's important that people, I think actually Laura said it the nicest is if it can slow down someone's diagnostic journey in some way. That's really important. And so getting a family to know what the implications of their diagnosis is important.
And I'll be brief. I mean, I think it adds to the other people. I think it's more for people I see in clinic that have had their symptoms for many years. I think sometimes they'll say, I have what I have, and sometimes it's trying to have a discussion about why you want to have genetic testing and bringing hope back to that person about where things are at today. And so that's already tied in, I think, to the other we talked about.
Are there other questions? Raise your hand so I can see you. Oh, way back there. Yeah.
Is there anyone that you don't want more genetic information about? Is there a way for you to say, these are people that we don't want to encourage? Maybe that makes it easier for us to look at those of us that should do more by telling us who you don't encourage to get more testing or more thorough testing.
Are you trying to say you're trying to determine those who may need additional testing versus those who already have a genetic diagnosis and don't need? How do you determine whether you need more testing?
I think the issue is that we want you to keep coming to clinic. And so I think that's the challenge. So that's a hard question to answer, because I may think of something new that I want to do, or I may need more genetic information, or I may find some newer genetic testing approach. And so once you've got a diagnosis, that's great, but our interaction should continue. And so I think that's something that happens is often patients get seen, and I think that's why many patients aren't genetically diagnosed, is because they were seen 10, 15 years ago, they were told they had limb-girdle muscular dystrophy, and they said, okay, I got a diagnosis, and they never came back. And so I understand, if we want to talk about barriers to care, coming to clinic and having me tell you, oh, you seem weaker, like, I get it, like, that's not very fun. But the issue is that if I don't know that you exist, and I don't know, I can't even start to think of those questions. So I really encourage you to come back to clinic, and it's a, I wish I could tell you it would be a great experience every single time you came to clinic, and I can't promise that, but it is important.
I can touch upon that question also. So just from the genetic studies that I work on, so it's just to tease apart two ideas: the idea of detection versus interpretation. So if you have variants of uncertain significance, it doesn't matter if you get a second genetic test, a third genetic test. The problem is not detecting those variants, it's the problem of interpreting those variants. Just to clarify to people, because yes, a different clinical genetic lab may have a different interpretation, but that all things being equal, the interpretation should be very similar between genetic labs for variants of uncertain significance. So it's not going to shed light on that in most cases, just getting another genetic test, because the DNA shouldn't shed light on that. And the other thing, just to point out, for a lot of the genetic tests that we're talking about here, the next generation sequencing tests, there are some things that it can't detect. So there are certain diseases, certain variants that it can't detect. So one is myotonic dystrophy and the other is FSHD. So we'd like for those people to go to a proper clinic, get examined, and for the proper test to be ordered, because it's very different to a lot of the mainstream tests, like say, for example, what VTA does and the Lantern Project. It won't be able to detect that. So, and that's why it's very important to come to clinic also And the last point is, and Dr. Wicklund touched upon this, is that not everyone with muscle weakness actually has a genetic disease. So some of those are autoimmune diseases or other causes. And so it doesn't matter how many genetic tests you get, it's not actually going to find the cause for you.
I wanted to make a point that if you had your genetic testing done a long time ago and you had a variant of uncertain significance, that variant may have been reclassified in the time from when you were first diagnosed to now. Either it's been determined that it's actually benign, or maybe determined it is pathogenic. So it's important to have a discussion every time you go back with your clinician and ask them, can you check and see if my variant has been reclassified? And that's what the GRASS group is doing. The Jane Foundation is trying to do that with variants in dysferlin. And so a lot is going on that something new could come up even if you've already had genetic testing. There could be new information as time goes on. So it says the time is up, but I'm gonna allow for one more question. I have one if no one else does. So I have a question because it came up when I saw Dr. Weil's results. When it comes back, because I get this question a lot, that it comes back and it has variants in multiple genes, and then somebody goes, oh my goodness, I have like 3 different muscular dystrophies because there's variants in multiple genes. So how does somebody know, do they have more than one? Is it only one of those? How does somebody interpret that? Especially when you get these big panels, you're going to have variants in more than one gene, and how do you sort that out?
Yeah. So you guys all know that this is a rare disease. The odds that you have 2 rare diseases is pretty low. So I would not make any assumption that if you had variants in 2 different classes of genes that you have 2 diseases. The likelihood is that you have one disease, and the other ones are just variants of unknown significance that are benign, that are unrelated to your current phenotype. Now, I say that knowing that there are exceptions to every rule, but in general, the diseases that we're talking about you likely have one genetic cause of muscle weakness for a limb-girdle muscular dystrophy, and those others are just noise as far as in the genome, so noise in the genome.
I can add one other comment. In a situation like that, sometimes it's really helpful to have a sibling tested because the siblings affected, they likely have the same change. If they're unaffected, what they have is probably not related. And so sometimes testing other family members can help resolve some of those confusing reports.
I guess to clarify, testing unaffected family members is just as helpful as genetic testing on family members. So it's not just, do you have a sibling that has a similar disease? It can be a sibling that doesn't have the disease. It's very helpful. And even if you're older, testing your kids, it's not just your parents. Any pedigree architecture that we can get is helpful.
So I'm sorry that's all the time we have. I just wanted to remind you, if you're interested in some of the tests, Dr. Kang's flyer here and the free genetic testing, there are flyers up at the front. You can come up and get them. We have plenty. And one other thing is I've been told that we have found a lost credit card. The name on the card was Walter Paul. If that is you and you lost your credit card, it's at the registration desk, which is down there.
And lunches are available. The box lunches for you to grab are in the exhibitor hall, and you can bring them wherever to have your lunch.
And the exhibits will be open during lunch, and the sessions start again at 1:50. So go enjoy your lunch, and we'll see you back at 1:50. Thanks, everybody.